Good afternoon, or rather good morning, everyone, depending on where you are in the world. My name is Pablo Chauke, and I'm a training and outreach coordinator based at HJ Barnett at UCT, or University of Cape Town, under the leadership of Professor Nicola Mulder. So one of my jobs as an outreach coordinator is to organize webinars. So there's a group of us. We are a webinar task force, and we make sure that we organize monthly webinars as well. This is the April webinar, and I'm super excited and looking forward to it. And it's been fun organizing it in the background with Sean Aaron, who made sure that we got in touch with Bhanu. And she was really just out there when I said, you are invited. And she was like, yeah, I'm here. So I'm looking forward to this very webinar because she's been very kind and enthusiastic for this webinar. So the theme is methods for variant discovery in microbes. And Bhanu is from the institute, or rather the Broad Institute of MIT and Harvard. This webinar is going to be for 40 to 50 minutes, so just a few ground rules. And at the end, we're going to have a 10-minute question and answer session. So just a few rules is to just say, please mute yourself if you're not speaking. If you have a question, raise your hand. There's at the bottom, or rather, if you hover over your name, you'll get options of reaction. So you can clap your hands, you can raise them, and other emojis as well. But I mean, I suggest that you kindly raise your hand if you have a question, or please wait for the end of the webinar. Right, she's going to speak for 40 minutes, and at the end, you'll have your chance to raise your hand and throw those questions to her. Alternatively, if the question is burning and you can keep it in, type it in the chat box to make sure that you don't distract her while she's speaking. Okay, thank you. So just a few lines from the abstract. I mean, it's a bit long, so I'm not going to read the whole thing because you're going to hear the talk now about what this talk is about. So the title is GATK for microbes, and part of the abstract says the detection of mutations in any microbial genomes is essential in understanding drug resistance, immune evasion, and other epidemiological characteristics of infectious diseases. In an effort to leverage the algorithms that have already become the standard for human genomic data processing, thousands of researchers have applied the Broad's Genome Analysis Toolkit, or GATK for short, to microbial variant discovery. However, this human-focused software may not currently provide the best results for all pathogens. For instance, genomic trouble spots, for example, repetitive loci, regions of high genetic diversity, translocated entirely absent regions, etc., are often ignored in human datasets as they represent a small percentage of the overall genome. In contrast, pathogen genomes are much smaller, and these trouble spots are proportionally more abundant. In addition, these same trouble spots often underlie clinically important phenotypes. For example, severity, transmissibility, resistance of antimicrobials, etc., that are immediate subject of investigation and thus cannot be omitted from analysis. To provide bacterial research community with robust variant calling methods, funded by the Chan Zuckerberg Initiative's Essential Open Software for Science program, they optimized the GATK to call short variants on bacterial genomic datasets. If you want to read the rest of the abstract, you should look at the ad that I send out. But now I'm just going to quickly read a bit, because I mean, she's done a lot. You can check her on LinkedIn or Google her to see, obviously, her illustrious CV as well, because she's done amazing for herself in the science career. Bhanu Gandham is a bioinformatics scientist, and she's very passionate about developing new and improved computational solutions to solve research challenges that the biomedical community faces. Before joining the Broad Institute, she has a well-rounded industry and academic experience working as a systems engineer at an IT company, a bioinformatics staff scientist in an epigenetic lab at Emory School of Medicine, and as a bioinformatics lead of exome pipelines at Veritas Genetics. At the Broad Institute, Bhanu previously led the bioinformatics support efforts for the Genome Analysis Toolkit, or GATK for short, a software package. In that role, she was responsible for helping genomics researchers adopt and efficiently implement the GATK tools. Closely interacting with the genomics community and having worked as a software engineer and as a scientist in academia and in industry, Bhanu has gained a unique perspective which helps her to identify gaps in engineering to support genomic research. It enabled her to see firsthand the need for bioinformatics support in microbial research, which inspired her to take on her current role as a computational scientist, leading development effort to build best practices for microbial variant discovery. amongst other projects that are aimed at solving biomedical research challenges in the community faces today. I mean, I can continue reading her bio, but I'm looking at the time, and I just want to say, Bhanu, thank you so much for taking time out to give this webinar. You can take the mic and start speaking. Thank you so much, Bhanu.
Thank you so much, Ravalo, for that amazing introduction. All right, let me share my screen real quick, and we'll get started. Are you able to see my screen?
Yes, Bhanu, we're able to see your screen.
Perfect. Well, hello, everyone, and thank you again, Paula, for that introduction. And I'm very grateful for this opportunity to present in front of this amazing group today. The goal of my talk is to share with all of you the GATK for Microbes project and the work that we have done in this project to develop improved methods for variant discovery in microbial species. But before I begin, I would like to thank my team at the Data Sciences platform at the Broad, especially Andrea and James, who have worked on this project with me. Special thanks to the bacterial and the fungal genomics groups at the Broad Institute, who have generously contributed and provided invaluable expertise to this project. I would also like to thank the Chan Zuckerberg Initiative, who funded this project. In this presentation, I will cover first a brief overview of what GATK is. I'll talk a little bit about what inspired and motivated us to develop GATK methods for microbial analysis. I'll provide details of the additions and improvements we've made to the GATK in order to optimize it for microbial genomes. And finally, I'll share with you how we are making this project available in the cloud and how that will help with scalability, portability, and collaborations. So let's begin with what is the GATK? GATK stands for Genome Analysis Toolkit. It is an open source software package. GATK essentially discovers variants after reads come off the sequencer. GATK has various tools for all major variant classes. For example, for germline SNP and indel discovery, we have the haplotype caller. For somatic SNPs and indels, we use MUTEC2. We have a suite of tools for germline and somatic copy number variation. And although still in beta, we have a set of tools for structural variant discovery, which covers germline structural variation. We're also going to develop tools for somatic structural variant calls, but that's work that's going to be done in the future. But that's not all that GATK does. In addition to all these variant calling tools, we also have tools for data pre-processing, post-processing of variant calls, such as filtering, call set evaluation, and variant annotation, and so on. In fact, there are more than 240 different tools in the GATK software package. And GATK has been in active development since 2009. The first version of GATK came out in 2010, and we are now on version 4, which came out in 2018. And as of version 4, GATK is fully open source. In addition to all the various tools that I was mentioning earlier for variant calling, GATK development team is also highly focused on ensuring that GATK tools are time efficient, scalable, and versatile. We also provide fully automated and reproducible GATK best practices workflows so that researchers can focus on their analysis without worrying about the computational complexities and aspects of it. GATK has a very large user community of over 75,000 users. And to ensure that our community is well supported, we have a dedicated community support team that answers users' questions on the online GATK forum, and we ensure that researchers are not blocked in their research work. However, there is one challenge. GATK was never intended for microbial genomes. GATK, in fact, was designed primarily for human data. And even though GATK was not optimized for microbes, we noticed that the community, the biomedical community, was already adopting it for microbial variant analysis. What you're looking at on the slide here is a timeline of all the papers that have been published on microbial research that use GATK for variant calling. One such example is SnapperDB. It is a database of bacterial variant information, and it is used by the England Public Health Department for pathogen surveillance. Here's another example of a paper that was published on the study of antimicrobial resistant properties of Enterobracter strains isolated from the International Space Station. Here's an example of a study on the outbreak analysis of Klebsiella pneumonia in Portuguese tertiary care hospitals. Characterization for Pseudomonas strains, public health risk assessment studies, and there are many other. Sorry, was there a question? I didn't quite catch that.
Sorry, that's someone who was distracting. Sorry about that. Continue.
No problem at all. I'm happy to take questions. Please let me know. And yeah, so like I was saying, there's all this amazing, important research that's being done using GATK, even though GATK was never optimized for it. And seeing how the microbial community was already adopting GATK for its analysis, we decided to collaborate with experts in the field of infectious disease research, internal and external to the broad, and work together with them to develop improved and accurate GATK methods for microbes. And that brings me to the three main goals of this project. First, we wanted to develop scalable, portable, interoperable, and reproducible software for efficient and accurate analysis. Second, we want to enable researchers to be able to study how microbes evolve, how they develop resistance and cause diseases. And for this, they need efficient tools to be able to look at the differences between strains on the nucleotide level with very high accuracy. And finally, we want to ensure that researchers are able to share with each other their data, the tools and their results in analysis, and be able to collaborate on a global scale. So let's take a step back and talk about what are some of the challenges with using GATK as it stands today before any improvisations for microbial genomes, and what are some of the challenges with using that. So first of all, it is the structure of microbial genomes. Microbial species can have variable ploidy, and many of them have circular closed genomes. While human genomes, on the other hand, which GATK is optimized for, are linear and diploid in nature. Second, microbial genomes are very highly diverse. For example, if you take any two strains of E. coli, they at the most, they share about 50% of their genes, while human genomes are about 99.9% identical. Also, microbial genomes are much smaller in size compared to human genomes. For example, bacterial genomes are about 0.1% of the human genome size. And therefore, sequencing and variant calling errors made with smaller microbial genomes are much more, it's much more important to deal with those errors than it is with genomes like human, which are much larger. So here is an overview of like the high level improvements we have made in GATK to optimize it for microbes. The first one is switching from GATK's germline variant caller to GATK's somatic variant caller. So going from haplotype caller to mutex2. During our investigations into looking at how the community was adopting GATK, we noticed that most researchers were using the haplotype caller for their analysis. But in our evaluations, we found that GATK's somatic variant caller mutex2 does much better with microbial genomes. And it makes sense because if you think about it, the rate of mutation and the high diversity in microbes is more similar to somatic data than germline data, which is why we decided to go with mutex2. Second, we are also focused on developing improved methods to increase the sensitivity of microbial variant calls. And finally, we also optimize GATK's filtering methods for better precision. So now let's kind of go into the details of some of these improvements that we've made in GATK. First up is the new Shift Faster tool we developed to optimize for the circular genome structure of microbes. So the problem that we're trying to solve here is essentially that when circular microbial genomes are sequenced, an artificial breakpoint is introduced to linearize their genomes for the ease of sequencing. So any reads that span this artificial breakpoint are lost for variant calling. And so any variants that might have been on the reads that span the breakpoint are lost to us. Which is why Andrea, who's a principal software engineer at the Broad Institute, developed a tool called Shift Faster. This tool solves the problem in 2 steps. First, it calls variants on one half of the genome that's away from the breakpoint. Then it shifts the reference faster by 180 degrees and calls variants on the second half of the genome. It then puts both of these call sets together and essentially rescuing any reads of variants that could have been on the reads that span the artificial breakpoint. Now let's talk about some other improvements we made in GATK algorithm itself for better microbial variant discovery. But before I do that, I want to give you a little background information on how GATK worked before we made our improvements. So GATK is a local assembly-based variant caller. Before the variant calling step, GATK uses alignment or pile-up data to identify regions of the genome that have evidence for variation. And this is called an active region. So first it identifies these active regions. Then it takes read information that overlap these active regions and creates a graph which represents all possible variant paths from the reads. And then by assembling the various paths in the graph, it creates a list of potential haplotypes. It then maps all the reads back to these haplotypes to determine which one of these haplotypes has the most evidence and hence generates genotypes, likely genotypes based on the haplotype information. And this is an overview of how GATK works under the hood without the microbial improvements. And this method has worked very well for human data. One of the biggest advantages of this method is that it is very, very accurate and sensitive to calling insertions and deletions compared to the traditional pile-up based variant callers. However, there are some blind spots to the GATK's previous assembly-based method. And here is one such example. What you're looking at here is an IGV screenshot. Many of you have probably are already familiar with IGV, but for those of you who are not, let me quickly explain what you're looking at, because we are going to be looking at a few other IGV screenshots in this presentation. So the first two tracks in this IGV view are variant tracks. It's basically just giving us information on whether a variant was seen in that particular sample or not. And then you have these three other tracks where you have read data. These read alignment data is basically all the reads aligned to the reference in that particular location. So this is essentially, you're looking at the pile-up data in these three tracks. So these bars that you're looking at, these gray bars, those are the reads that are mapped in this region. These empty bars that you're looking at are essentially mapping quality 0 reads. And all of these colored bases are bases that are mismatched compared to the reference. So basically here, it's saying that there's a C mismatch while there should have been a different base from which you see in the reference. So from the Illumina reads, you can see that it looks like there are actually 4 mismatches in the region. However, the nanopore and the PacBio long reads are showing us that there's actually a huge deletion event in this region. And haplotype caller wasn't able to identify this deletion event. However, GATK for microbes was able to successfully identify this deletion. So why was haplotype caller not able to pick up on this deletion? And the reason for that are dangling ends. So as you can see, there are these mapping quality 0 reads on the other side of this variant information. And so basically, when GATK is creating its assembly graph, it doesn't have enough information on the other side to create a continuous graph. And therefore, the graph breaks up and creates these dangling ends which don't connect. And we lose any variant information that are on these branches, and essentially losing that variant call from the sample. To solve this problem, James Emery, who's a senior software engineer at the Broad Institute, wrote a piece of code to help rescue these variants. And which is why we can now see this deletion event in the GATK for microbes improvements. So one other thing that we did was we looked at, this is one example of the false positives that we weren't initially able to capture. We looked at many other such false positives. And we realized that a pattern was starting to emerge from all of this. And the pattern was that most of the variants that we were not able to call with GATK's assembly-based method, we were in fact able to pick those up using other pile-up-based tools like Freebase. And that essentially brings me to my next improvement. Given how we were seeing these advantages with both local assembly-based method and pile-up-based method, we created this hybrid GATK, which combines the advantages of both of these methods. If you remember, I mentioned earlier that the first step in GATK's assembly-based method is to look at the alignment or pile up data and identify the active regions. Previously, after it finished with identifying the active regions, it would essentially like throw this pile up data information away. However, in our hybrid method, we save this pile up information, and we use this pile up data to generate additional pile up haplotypes, which we then throw back into the mix with the other assembly-based haplotypes. And then we allow the reads to decide which one of these haplotypes has the most evidence. This hybrid method has drastically improved the sensitivity of GATK on microbial data. And let me show you some examples of these improvements. So here's an example of a set of clustered events, which we weren't able to see with the assembly-based method, but the pile-up-based method was able to rescue these variants. Again, in this IGV view, the first 3 tracks are variant tracks, and the last one is alignment data from Illumina reads. The first track here is the truth data. Truth is basically a list of high confidence variants that are used for evaluation. In this case, this truth data was generated using assemblies from nanopore and PacBio long read sequencing data. And we also, and a lot of extensive manual curation was done to generate this high confidence list of variants that we call truth. This truth data was generated by Ashley Earle's team. Ashley is the senior group leader for bacterial genomics at the Broad Institute. And she's one of our panel of experts for this project. And we're very grateful to her and her team for helping us with this project. So we use this truth data to evaluate the impact of the improvements we are making to the accuracy and the sensitivity with which we are calling these variants. The second track here is the GATK's assembly-based haplotype caller, and you can see that it doesn't call any variants in this region. And then the GATK, the hybrid GATK, or the GATK for microbes method, was able to rescue all of these variants. So the reason that haplotype caller wasn't able to identify these variants is because when you see such clustered events together, the assembly graph becomes, the local assembly graph becomes very large and has many branches, and GATK is not able to process this large graph, and therefore it completely drops it, and we lose that variant information. However, the pile-up based method is able to see and identify and process this data. Here is another example of a variant, a true variant that is nested between these soft clip bases, which haplotype caller was not able to identify, but GATK for microbes improvement was able to. What you're looking here is a very messy soft-clipped region. What I mean by soft-clipped are soft-clipped bases are essentially bases that are flagged by the aligner to let us know that the aligner wasn't sure which region of the genome these bases align to. But because they're part of these other reads, part of these reads with good alignment, it places it here, but also it flags it appropriately and says it's unsure of where it should go in the genome. And because of this high diversity in microbial species, we see a lot of such true variants nested in soft-clipped bases, and we see that a lot in microbial data. All right, so I've talked about what are the improvements we've made in GATK. Now, let me show you some evaluation results and comparisons of these of GATK's improvements with other tools. So the first evaluation result I'm showing you is was done on simulated bacterial data. We used E. coli species here. We used the sample reads were simulated using neat tools. The reference we're using here is the CFT073 strain. The tools we've used here for evaluation are Pylon, haplotype caller, freebase, and we're comparing it to our GATK for microbes hybrid method. And the reason that we're using these other three tools to compare it with is because Pylon is a whole genome assembly-based variant caller. Haplotype caller is a local assembly-based variant caller. Freebase is a pilot-based variant caller, and GATK for microbes is a local assembly and pilot-based variant callers. And so we thought this would be a good mix of algorithms to test our improvements against. And so as you can see, that the precision for all the four tools are almost similar, but GATK for microbes does much better with sensitivity and recall compared to the other tools. But the results that you're seeing here are on simulated data sets. Simulated data sets are great because with that, you know exactly where all the variants arise from and where the location of all the variants are. However, simulated data cannot capture the essence and the complexities of real data, which is why I'm also showing you some results, evaluation results on real fungal data. The species here we're using is Candida auris. The sample is strain B11221. The reference is strain B8441. This test data was generated and shared with us by Christina. Christina is a senior group leader of the fungal genomics group at the Broad Institute. And Christina is also a part of the panel of experts for this project. And we're very grateful to her and her team for helping us and advising us on this project. So the results that you're looking at here is for this real fungal data. And as you can see that the recall for GATK for microbes is much higher than the other tools. However, we're not doing as well with precision. All the other tools are doing better than GATK for microbes for precision. And this is work that we're still doing. We're still working on improving our precision rate. And with all the improvements we made to the GATK for microbes method and the hybrid method, but while we were increasing the sensitivity and recall, we were also losing precision. But like I said, we are going to continue working on this and try to make sure that our precision is comparable. All right, so now let's switch gears a little bit and talk about our final goal for this, for the GATK for microbes project, which is essentially to enable global scale collaborations between researchers and also allow researchers to be able to share their data and analysis. To do this, we are using cloud-based methods. So the way traditional non-cloud-based methods worked was you would have a central repository that holds all the data, and each researcher would copy data onto their local storage to analyze it with their own tools in their own custom environment. But there are many drawbacks to this traditional approach. One, data sharing is much harder to do when you're making copies of it on your local computers and your local systems. And you now have multiple copies of the same data, and this also increases the storage costs. And because you're making your own copy and then you're using your own custom tools and environments, results may not be as reproducible. Which is why the cloud-centric approach, on the other hand, helps with collaborations, because with the cloud approach, you have a central repo in the cloud, which researchers use to access data but not copy from. And you can analyze data together, collaborating in a virtual environment. And this essentially opens the door to be able to, excuse me, immediately share data, tools, the versions of the tools in Docker containers, all of which means that this would help make results more reproducible, therefore making collaborations much more easier. This is where Terra comes in. Terra is a cloud-native platform developed by the Broad Institute in collaboration with Microsoft and Verily. Terra is a secure, scalable, open source platform for biomedical researchers to access data, run analysis tools, share their data and results, and collaborate on a global scale. The platform allows you to do all aspects of your research project in one place. Terra has libraries that include public and restricted access datasets. It has workspaces where you can upload your own data, your own tools, workflows, and share that with your collaborators easily. It also has two modes of computations. So not only can you run your workflows in Terra, but it also supports interactive analysis. So it supports Jupyter Notebooks, Galaxy, and RStudio, so that in addition to running your workflows and data, you can also evaluate and review your data using these interactive analysis tools. And finally, we have made the GATK of microbes available in Terra. What you're looking at is the GATK for microbes best practices automated pipeline. It has three parts to it. It has the data pre-processing steps. It has a section on variant calling, and then finally evaluating your call sets against a true data set. All of this we make available in Terra by putting our tools and their versions in Docker containers, so that we control the versions that are used by collaborators who are sharing this data and tools and workflows. And we make all of that available in Terra, which essentially makes it very easy to run our analysis and share data and results. This is the link to our to the GitHub repo of this project. There's a link to this workspace in Terra in this GitHub repo if you're interested. And that brings me to the end of all the work that we've done so far. The next steps for GATK for microbes is, one, we want to expand GATK for microbes to all pathogens. We want to be able to apply it to bacterial, viral, fungal, and parasite protozoan data. And we have a lot more work to do to make it to enable GATK use in all of the pathogens, but we're working towards that. Another application that we want to work on developing is microbial longitudinal analysis. We want to develop tools to be able to look at and to pick out the differences between strains in say an outbreak. We want to be able to do some strain level longitudinal analysis. We also are working on developing an automated machine learning based approach for parameter optimization of GATK workflows. What that essentially means is when you're using any workflow, not just GATK workflow, it has these list of parameters that users can tweak. And this list of parameters always they come with default values that are assigned by the authors of the tools based on their experience with the data and with the tools. And we almost always use these parameters with the default values. But to be able to tweak these, you need to have prior knowledge of the data and the heuristics of it. But we want to develop an automated way to do that. We want to develop, we've taken inspiration from a machine learning method to tweak a list of parameters and give you the most optimum values for each of those parameters so that you can use that in the most efficient way in your analysis. And that concludes the talk for today. I want to thank this amazing group who have helped with this project, and none of this work could have been done without their help. And I'm very, very grateful to this group. And I've shared some links if you want more information on GATK or Terra or the GATK for Microbes project, please feel free to reach out to our team using these links. Thank you, everyone. And that concludes my talk for today.
Okay, this was amazing. Thank you so much, Bhanu. I saw some familiar faces on acknowledgement, some people that we've worked with before. So it was nice to see. There are a few questions already. So this is question and answer time and discussion. But firstly, thank you, Bhanu. This was amazing. I'm just going to field some of the questions from the, David says, this was great. Several quick questions from me. So this is David Tab, who's in France. Is ensemble bacteria an appropriate source for reference annotation? Does Terra have servers in Europe? Getting data from Africa to cloud servers in the USA is slow. Is alpha status of the GATK for microbe software barrier to production use? Will old Java version dependency resulting from use of Spark soon be resolved? Okay, these are multiple. I hope you're writing them down. Thank you, David. Is gzip standard way to compress FASTQ files for long-term storage? So those are all from David Tab. And there was a question from someone called Genetics at One, but I think you can answer David's questions first, and then we can move on to the next question afterwards. Thank you.
Thank you. Thank you for those questions, David. Let me tackle them one at a time. Is ensemble bacteria an appropriate source for reference annotation? We haven't gotten to the part of reference annotation yet. We're still working on tackling the variant calling aspects of it. But not from my personal experience, but I've looked this up, and yes, I think ensemble bacteria is an appropriate source for reference annotation, but I don't have any more information on that, unfortunately. Moving to your next question, does Terra have servers in Europe? Getting data from Africa to cloud servers in the US is slow. Yes, I believe I've heard that getting data from Africa to cloud servers in the US is slow. I'm not entirely sure if Terra has servers in Europe. I think it's on the Terra website. There's a list of different servers all around the world. I believe there is, but I will need to check. For for any of these Terra-related questions, I would encourage you to post on the Terra community forum. They will provide you with all the most accurate and up-to-date details with you. And I think to answer another question, Terra currently uses the Google Cloud platform. And I know that those servers are only located in some countries, but Terra is now going to also be expanded to using Microsoft Azure. And so that will open up the use of many more servers around the world. And that'll help with access of Terra in many more other countries. Is alpha status for GATK for microbes software a barrier to production use? At this point in time, yes, it is. I only say that because we are still making more improvements, and then in the coming weeks we will have a new release, a production level release, and however, right now, because we are still testing on a lot of data, I am not entirely sure if it's ready for production use. Will the old Java version dependencies resulting from use of Spark soon be resolved? I think this is probably for a different, this question is probably for a different team on the GATK development side. And I don't work on the Spark development as much. But again, if you, for these questions, you can either, I've put my email address on the slide that I'm still sharing, or the GATK community forum. You can reach out to us on either one of these links, and I can get you more information on exactly what of the issues that you're mentioning about Spark with the old Java version, and we can help you get more information on that. Is gzip the standard way to compress FASTQ files for long-term storage? I think for now, gzip is something that's very commonly used for long-term storage of the FASTQ files. I hope I answered most of your questions, David. These are great questions.
David, you can unmute yourself. I've given you rights now. You can speak. I see you nodding your head. You can say yes. You're able to unmute yourself, but he's nodding his head. So seems, are you speaking? Wait, okay, let me unmute you. How do you, because you're chuckling away. Why are you not asked to unmute? David, unmute yourself. Okay, he's saying something. I don't know why he can unmute himself.
There, magical, great. Yes,
oh.
I tried unmuting and it said that the host is disallowing unmutes. So I just want to say thank you. That helps a lot. This is a very new field for me, and I really appreciate your answering the basic questions. Thank you.
Of course, my pleasure.
Okay, thank you, David. Thank you for your answers, Bhanu. There's some question from genetics@1. I don't know who that is. There's two, and I think this is very specific, so I might ask them to unmute as well, because I think this question came around your 4th, 5th slide, and this is the question. Thanks for the great talk. How did the use of GATK affect the results of those studies? Were they in fact valid? And following that, they say, is there a need for reanalysis? That's from genetics@1. I don't know if genetics@1, you want to say specifically what part of the presentation you were asking about? Genetics@1? Let me, hello? Let me ask them to.
Hey, yes, Pablo, it's San James, actually.
Hi, your name. You should have named yourself properly.
Yeah, thanks, Bhanu. This is really great. I think it was on the slide about the unintended use of GATK, and you kind of showed a number of studies that were done prior to the improvements, and I think that from the rest of the presentation, it might be quite interesting to reanalyze these studies and see how much different the results would be. But of course, earlier on, I was just asking to find out if indeed the results presented in these studies were valid.
That's a great question, and thank you for that question. I think, first of all, we are going to do recreate some of the published data and see how the improvements are, either helping or how they're helping with the accuracy with the new improvements. But another thing to note, and thank you for bringing this up, because that brings me to this next point, which is, as you can see, even I don't believe that with these new improvements, we're not trying to say that the previous methods were not accurate. If you look at the precision levels on both the simulated data and on the real data, you can see that the precision levels, and using the haplotype caller, the version of GATK before the improvements, they're still very high. What the difference that it will make is that a lot of variants that we were missing before, now we will have more sensitivity for it. So it's not that what it was already capturing was wrong. It was just there's much more there to capture. And with these improvements, we're going to increase our sensitivity.
Sure. Makes sense.
That's a great question. I hope I answered that.
Yes, you did. Very well. Thank you.
Awesome.
Thank you so much for that. Genetics at 1. We still have time for about 5 minutes for comments and questions. So please do go ahead, either type in the chat box or raise your hand, and you have the right to speak now. So raise your hand, and obviously, if I pick you, please do go ahead and ask a question. I'm just busy checking to see if there are any questions in the chat. I've also pasted the email address for everyone in the chat box to see, and I'll also be able to share slides. And we're recording this presentation, so it's live on Facebook. You can go on Facebook and check it out, because it's going to be there after this presentation, or alternatively wait for the email I'm going to send with the YouTube link. And I'm going to put it on social media, and I'm going to email it as well. So if you're waiting for that, for those that missed the first few minutes, don't worry, it was recorded, and you'll get all of that recording as well. Okay, 3 more minutes.
I also see that Andrea has posted and shared a lot of links in the chat with all of you. Thank you for doing that, Andrea. And yeah, please feel for anyone who has any more questions, outside of this presentation, please feel free to reach out to us using any of the links that you see on the screen right now.
Okay, thank you. David was clapping his hands as well, and I'm really grateful for this, Bhanu. This was amazing. Thank you so much for your time. I appreciate it. And yeah, and other people like Andrea. Thank you, Andrea. Okay, so to the rest of you, see you next month in May for a different webinar with a different speaker. I'll send out the ad as the time approaches. But yeah, Bhanu, this was amazing. Thank you so much. Enjoy the rest of your day, everybody. Goodbye.
Thank you for this opportunity. This is a great audience, and I'm delighted I got the opportunity to present to all of you.
Thank you, Bhanu. Oh, and people should follow you on Twitter, even though you're not active. People must, you have a Twitter account, so people must follow you there as well if they want to get updates, or follow the GATK_dev account on Twitter as well. And they obviously regularly post links and updates on their toolkit. Thank you.
That's right, and I will try to be more active on Twitter. But yes, the GATK_dev is handled by our marketing team, and so they will keep you posted on all the latest updates with GATK. And we also have a blog on the GATK website, so you can follow that as well to get more information on the recent and the newest developments in GATK.
Okay, that's amazing. Thank you. We'll check it all out. Sean, saying thanks, Bhanu. That was a great talk, and yeah, thanks to him for making this happen. So thank you so much. Enjoy the rest of your day, everybody. Goodbye.